Good afternoon, my name is Sibylle Mellinghoff, and today I would like to present you a case of a patient with Aspergillosis and Mucormycosis co-infection that was treated with an immune checkpoint inhibitor. It is a cooperation project of the University Hospitals of Cologne and Munich. Invasive fungal infections are serious threats to immunocompromised patients, and especially mucormycosis is a disease with high morbidity and mortality despite treatment options by antifungals and surgery, as we can see in this graph. Mortality remains significant, and therefore novel treatment options are urgently needed. Preclinical data suggest the benefit of immunotherapy for invasive fungal infections, and especially immune checkpoints and in their inhibition seem to play an important role. We have seen such data from mice to date, and also one study from patients with candidemia, where all 27 patients showed an immunophenotype consistent with T-cell exhaustion. Therefore, colleagues from Belgium treated a young patient with mucormycosis after a polytrauma successfully with nivolumab. The patient recovered, and they reported that case in 2017. Against the background of these data, we treated the first patient with a hematological underlying disease with nivolumab. That patient was 51 years old, and she had the first diagnosis of AML in 2015, the first relapse in 2018, then had allogenic stem cell transplant, and the second relapse in 2019 last year. I would like to go a little bit more into detail from that point on. I know this figure is quite a complicated one, but I'd like to guide you through that. The patient presented with fever, loss of affectation, and sinusitis complaints. In the CT scan, we saw the sinusitis, and microbiologically, we diagnosed mucormycosis and aspergillosis co-infection. The patient then received amphotericin B and several surgeries within the next days, but however the infection and the clinical situation worsened. Therefore, amphotericin B was combined with isavuconazole and then further dose escalated. But the disease progressed even though, and the CT scan some days later showed bone destruction and spread into both orbits now. Due to lacking treatment alternatives, we offered the patient nivolumab as a treatment option at that point, and she agreed. So 2 weeks after the diagnosis of the invasive fungal infection, the patient received nivolumab for the first time. We combined nivolumab with interferon gamma, as did our Belgian colleagues, and 2 weeks later we saw the first signs of clinical improvement. The patient didn't have fever anymore, the signs of sinusitis regressed, and no further surgeries for hemorrhagic packing and debridement were necessary from that time point on. The patient received nivolumab every 2 weeks from now on and received 4 doses in total. Interferon gamma had to be stopped due to injection reaction after 2 cycles. Even affectation recovered 2 weeks later, and another CT scan showed regression of sinusitis signs, and only a little bit of swelling remained. After the 4th doses of nivolumab and persisting neutropenia, AML progression was diagnosed in a bone marrow biopsy, and the patient was discharged at that point. 4 weeks later, she didn't have any signs of sinusitis or fever. In the meantime, she presented at the emergency room with septic shock and denied any further treatment and died. To gain further insights into the immune response during treatment with nivolumab, we analyzed PBMCs from our patient, compared them to healthy controls. PBMCs were analyzed before treatment, under treatment, after 2 weeks and under treatment after 5 weeks. In both activation as well as maturation molecules, we saw significant changes, as you can see here, and also co-inhibitory markers were measured, and as expected, we saw before treatment an increase in these markers in CTLA-4 and PD-1, which are now shown as an example, and a decrease under treatment. To conclude, we here present the first patient with invasive and fungal disease and a hematological underlying disease who was successfully treated with checkpoint inhibition. And as seen in further analysis, the lack of immune defense was thereby targeted and possibly led to restored T-cell function. However, we do not know how specific this restored T-cell function is, and also we do not know the effect on the underlying AML. Indeed, this is a single case and needs evidence by further investigation. In case of any questions, please do not hesitate to contact me at this email address. Thank you very much.